So I did it again, and the stop button instead of the top side arrow that's right next to it. So what we just finished was we're talking about codons. We're talking about translation. The information in the mRNA is in sets of 3 nucleotides. Those nucleotides are called codons. This is where I wanted to go next. This is a picture book that is demonstrating or illustrating something that we very commonly call the genetic code. All organisms share the genetic code. It's very, very similar among all different organisms. And the way you read this chart is that this side shows you the first base in the codon. These letters show you the second base in the codon. And these letters show you the third base in the codon. So if you're looking for G, U, U, first you find the G, then you find the U, and here you find the U, and where all 3 of these come together is the G, U, U. And you can look next to it to see which amino acid corresponds to that codon. The proteins are these strings of amino acids. One codon dictates which amino acid is going to be put into the protein. That codon came from the DNA. Putting those amino acids together is called translation. And so this is a figure that shows you that. So here's the region of the DNA. They're showing it to you in blue because it's DNA. They color code it, and that's actually pretty helpful to try to keep in mind what's going on at each point. And so we use our DNA base pairing rules to make an RNA copy. Now, they're showing you the template strand and the RNA strand. They're showing you what I was pointing out when we looked at this, which is the same information pretty much is in the DNA gene and the RNA strand, except where you have a T in the DNA, it becomes the U in the uracil. So this figure is not showing you base pairing. It's showing you that the information in those 2 strands is the same. They're showing you the same thing that I was showing you with the figure . So you can either use a DNA template strand to make a copy, DNA, replication, or you can use that same DNA template strand to make an RNA. That's transcription. But if you look at this RNA strand and this DNA strand, they are identical, except when you have a T in the DNA, it's replaced by U in the RNA as uracil. And so that's what they're showing you here, is it has the exact same information. The information in the gene and the information in the RNA is almost exactly the same. And then we take these 3 nucleotides as a single unit. Is called a codon, and it codes for 1 amino acid. These 3 code for this amino acid. These three code for this amino acid. These three code for this amino acid. These three code for this amino acid. And how did we know that? We used this figure. So UCA on that previous figure was a serine. UCA is a serine. So these amino acids come directly from that genetic code figure. Now this is happening on the ribosome. There's two subunits to the ribosome. There's a large subunit and a small subunit, and they come together to form this ribosome complex. And there's a tunnel through the interior, and the RNA can string through the interior and assemble a polypeptide chain, and then thread that polypeptide chain out. And we'll look at an animation of it. So the rRNA and tRNA help to translate or do translation. Ribosomes and transfer RNAs interact to translate the mRNA into a string of amino acids that we call a polypeptide. And there's two subunits of the ribosomes, the large subunit and the small subunit, and they contain ribosomal RNA and structural proteins. During translation, one large subunit and one small subunit come together as a ribosome. They form around an mRNA molecule. The ribosomal RNA reads the mRNA and acts as an enzyme to form peptide bonds between the amino acids to make that growing polypeptide chain. So the ribosomal RNA forms part of the ribosome. The tRNA has really got an important job. It brings the amino acids to the ribosome, and it does so in the order that it needs to, according to the mRNA, according to the genetic code chart. So how does it do that? It has an area called the anticodon that is made up of RNA nucleotides that can base pair with the codon in the mRNA. The anticodon is a set of 3 nucleotides on the transfer RNA that base pairs with the codon on the mRNA. The anticodon is a set of 3 nucleotides in the tRNA that base pairs with the codon in the mRNA. I said that twice because that can be kind of a complicated sentence, but it's kind of important. So the tRNA, I have a picture of one. The tRNA is an RNA. So it's a long, single-stranded chain of these RNA nucleotides all strung together, and it's kind of all wound around itself. And there's 2 parts of it that are important. One end, it attaches an amino acid to itself. And at the other end, it has 3 nucleotides that are important that are called the anticodons. And they can base pair, form hydrogen bonds with the mRNA codon. So on the mRNA, there's going to be 3 nucleotides that form the codon that are going to match to this, that are going to be U, G, G. And they'll form these hydrogen bonds. And if you look on the chart, because this all comes from that codon chart, that figure 7.5 codon chart, If you go back to the codon chart and you look up UGG, the amino acid is tryptophan. And the amino acid is tryptophan because there's this structure called a tRNA. At one end, it can match up with the codon, and at the other end, it has an amino acid attached. So there's 3 stages to the translation process, and they're shown in your book in the image that we're going to look at next. Initiation starts translation when the mRNA joins with the very first tRNA and the ribosomal subunit. And then the process elongates because one after another the tRNAs come in, and which ones stay are the ones that base pair with the mRNA, and so their amino acids get strung together to form the polypeptide. And the last step is something called termination. You may have noticed on that chart there were 3 codons that didn't have an amino acid associated with them. There was a stop sign next to it. There are 3 codons that give the signal to stop translation. When the polymerase encounters one of those stop codons, the whole thing kind of falls apart, and that ends the formation of that polypeptide. So I want to go back and review some of this that we've looked at, because we've looked at transcription and translation, and we said some things in the beginning that might make a little more sense now that we've gone through all of the parts. Also, I accidentally hit stop in the middle of that, so if you're trying to go backwards now that we're at the end, that's going to be kind of difficult. So I want to go back through that transcription and translation piece. So we started from this slide. We said DNA has all the genetic information needed to make the protein. And that genetic information consists of sequences of bases. So we have genes that have all of these bases in them, and a gene dictates an RNA protein product. And then we have this sentence: the information is in the order of the nucleotide. When we looked at that chart, the codon chart, that gave us the clue about this sentence. The information is in the order of the nucleotide. Because when we look at the codon chart, all of these codons are 3 amino acids in length. So what determines the different amino acids is not the number, because they're all 3. What determines this versus this is not the first letter, because the first letter of all of these is a C. What determines it is not the second letter, because all of these start with AG. What determines it is the 3-letter sequence. The 3-letter sequence of nucleotides determines the different amino acids, determines which amino acids will be strung together. So this sentence, the information is in the order of the nucleotide. The order of the nucleotides determines which amino acid gets added to the polypeptide to form that protein. The 2 steps are transcription, DNA to RNA, and translation, RNA to protein. And so when we do these steps, we get gene expression. We take the DNA and make it into an RNA, that's transcription. And then we take the RNA and make a protein, and that's translation. And the way we do it, by using the base pairing rules. The base pairing rules are used at every step through this process. By using the base pairing rules, we are able to keep the information the same in all these different products as it was in the original DNA molecule. So even though DNA can't leave the nucleus, and these things are happening in the cytoplasm, the same information is in the cytoplasm as was in the DNA. So that same information is being put into a protein. So the first step was transcription. The DNA is in the nucleus, but we make proteins on the ribosome. So how does that work? We transcribe the base sequence, the nucleotide sequence of the gene, into an mRNA. And the mRNA carries that message to the ribosome. We had to introduce some changes to our base pairing rules here. This is the figure that we use to show the difference between RNA and DNA. And we also had to talk a little bit about how there's no E in RNA. This instead is U for uracil. You notice that we abbreviate these names of these nucleotide bases by single letters. So transcription uses an enzyme called RNA polymerase. RNA polymerase binds to a DNA sequence called the promoter. It unwinds the DNA, it fills in the RNA strand, and that new mRNA is a copy of the gene. And we went back and we looked at how there's exact same information between that DNA and that gene in that previous figure. This is here from your book that shows the transcription bubble and the filling in of the RNA. And when you look at the letters, you can see how the information gets transferred from the DNA to the RNA. And this can be happening all at one time. And that's what this figure shows you. So that's the transcription piece. There are several movies and animations that are in the Links and Animations tab in Blackboard that show the transcription piece. And there are several that show translation also. So translation starts with that mRNA that we made in the transcription step, and it has the same information that was in the original gene. It leaves the nucleus, goes to the ribosome, and then basically we translate the message into a protein. And how do we translate that message into a protein? We take every 3 amino acids, we look at the codon chart, and we see which amino acid they code for. So we can do that same thing looking at a DNA sequence. And the ribosome can do that same thing looking at a DNA sequence. And it's based on that codon chart, on using that codon chart, all organisms have a codon chart. So in the vegetable suite, we're looking at microorganisms. We're looking at antibiotics that fight against microorganisms. And one way to keep a microorganism from growing prevent it from making proteins. We can prevent it from making proteins by blocking this transcription and translation step. So here's the code, the figure from your book. Translate RNA to amino acids by using that code. We have two additional pieces of RNA that we talk about, tRNA and rRNA. They help form the ribosomal subunit, and they bring in the next amino acid. tRNA has two attachment sites. It has an anticodon, the base pairs with a codon, and then there's the amino acid binding site, and it binds the amino acid specified by the codon. Which amino acid is that? That depends on the chart. You have to look at the chart to figure out which amino acid. There's 3 stages of translation. And then here's the figure that shows the translation process. So you get 2 amino acids, 2 tRNAs right next to each other, with their amino acids next to each other, and you form a peptide bond. Then this tRNA goes away, the next one comes in, and you form a peptide bond. This tRNA goes away, the next one comes in, and you form a peptide bond. And what you've done is you've taken the information in the mRNA and translated it into a sequence of amino acids, and you've used these tRNAs in order to do it. So if you look really closely at the figure in your book, it gives you these nucleotides, these letters, these bases that are forming this particular amino acid sequence. We can practice this. Okay, so we do G, G, Q, G, G, U, G, U, U, A, G, G, G. AUG. AUG is one codon. GUG is the next codon. UUA is the next codon. GGG is the next codon. So now you go back to the codon chart, figure 7.5. It's on page 120 in your book. AUG. We're going to figure out which amino acid. So you look up A, you look up U, you look up G. Or you can just scan around until you find it. And you find out that it's a methionine. We abbreviate MET. Yes, you can abbreviate. That's not a problem. So then you can look up GUG. And when you scan around after you look up GUG, you find that it forms a valine. When you look up UUA, you find that it forms a leucine. And then when you look up GGG, you find that it forms a glycine. And that's exactly what they show you here. They show you those 4 amino acids that we just looked at, and they came from this sequence right here. And they were put together by this mechanism. The tRNA comes in, you form a peptide bond. The tRNA comes in, you form a peptide bond. The tRNA comes in, you form a peptide bond. Now, at the end of chapter 6, it mentioned mutations. Mentioned sometimes what happens when you make a DNA replication is that you get an error, and that can lead to a mutation. Since the order of the nucleotide determines what amino acid gets put in place by that codon chart, then if you change the nucleotide in the codon, you could change the amino acid in the protein. We said in chapter 6 that mutations are permanent changes in the nucleotide sequence of the DNA. It can alter a gene product because it could change the nucleotide in the codon and could change the amino acid that's in the protein. We have lots of examples of mutations that affect proteins that have harmful effects. An example that your book gives you is a mutation that affects the hemoglobin protein, makes it not able to carry as much oxygen. Now, there's different types of mutations. A deletion is when we lose a base pair. An insertion is when we gain an extra base pair. And that causes all of the amino acids after that may be altered, because if we're looking at every 3, suddenly when we lose one, and we look at every 3, now we've shifted. Or it can just be a substitution when the single base pair changes. It's not gained or lost, it just changes. And there's some figures in your book that show you some examples of these. So this figure 7.9b shows you part of this hemoglobin protein. Here's the DNA, here's the mRNA, and here's the resulting amino acid sequence. So this is the normal protein. If we get a single base pair deletion here, PGA, GGA, CTC is what it should have been in the original. We get a deletion here. This has changed the amino acids that come after it. The outcome is a disease called thalassemia, where their bodies can't make that hemoglobin. They don't have enough hemoglobin, and they don't have enough oxygen. Even adding an additional can change the reading frame and can change the amino acids that are added. This changes just a single amino acid, but this protein can't fold properly, so it even changes the whole shape of the red blood cell and causes the red blood cell to sickle and can cause sickle cell anemia, if you've ever heard of that. And they give more of an example in figure 7.10 about sickle cell anemia in your book. Most mutations are from not repairing those errors that DNA polymerase encounters during DNA replication, but there's other things. We can be exposed to radiation or chemicals, or even sometimes diseases that can cause mutations. Ionizing radiation breaks chromosomes. So here is a karyotype analysis where you can see broken parts of chromosomes, and that can cause mutations. UV light results in something called thymine dimers, where instead of base pairing with the A's and the T's, the radiation causes the T's to bind with each other. And then when the DNA polymerase comes through here, it doesn't recognize what we're supposed to be. And it just guesses, and it just sticks something in there. Maybe it guesses right and sticks in a T. Maybe it guesses wrong and sticks in a C or a G. So those are some ways that UV light can cause radiation damage. Excess amounts of UV light that cause excess amounts of these thymine dimers can sometimes lead to skin cancer. Some synthetic chemicals cause mutations in DNA. For example, cigarette smoke sticks extra atoms to the bases in the DNA, and so the hydrogen bonds don't line up. And so that can cause mispairing during replication and can cause a lot of mutations that way. Now, the last thing that I want to mention is a couple of terms that we can talk about as we talk about these cells growing. Cell differentiation is a term that means in the process of cells growing, sometimes they become different types of cells. They become more specialized cells. This is not like cancer. This is not uncontrolled cell growth. This is a part of normal cell development, that a cell becomes a very specialized type of cell that fits into certain tissues and certain organs. So all of our cells have the same complement of chromosomes. They have the same genes on their 46 chromosomes. But different cells express different genes. Remember that term, gene expression? Gene expression is when the DNA becomes an mRNA that becomes a protein. We say that gene is expressed. Different cells express different genes. And cells differentiate or become different when they start expressing a different set of genes than other cells. And so controlling gene expression is the basis of differentiation of cells. You can control gene expression by things like transcription factors. They bind to DNA and affect gene expression. And so if they're starting transcription, then they can affect that process. Now, there's even some antibiotics that use transcription factors as their target. So when you're thinking about an antibiotic to use for the discussion board, you can, as I mentioned in the discussion board in an answer to a question, you can Google transcription antibiotics, you can Google translation antibiotics, but you can also look at what antibiotics might bind to or regulate transcription factors, which are the proteins that help transcription take place. So this is a summary that kind of ties together both Chapter 6 and Chapter 7. So when a cell is dividing, it needs to copy its DNA so that it has enough DNA to make 2 daughter cells. And it does that by replication. When a cell is doing different cell functions, it needs proteins, and it makes them by transcription and translation. Both DNA replication and protein synthesis rely on our base pairing rules, but if anything involves the use of RNA, remember the RNA rules are a little bit different. The Chargaff rules for RNA replace thymine with uracil. So my hope is that that kind of explains some of the material for Chapter 6 and 7. As usual, any questions that you have, you can post on the discussion board, and I will try to get those answered for you.